Having learned the development of neural tube, we will now discuss the histogenesis, means development of the different types of cells of neural tube. The cells of neural tube are derived from the wall of the neural tube, whereas lumen of the neural tube gives rise to the ventricular system of brain and central canal of the spinal cord. So at first, the wall of neural tube is lined by tall columnar neuroepithelial cells which extend from internal limiting membrane to the external limiting membrane. Afterwards, the neuroepithelial cells assume pseudostratified appearance, so the nuclei of the different cells acquire different positions in the wall of the neural tube. Continued proliferations of the proliferation of neuroepithelial cells divide the wall of neural tube into three zones. From within outward, inner ependymal zone, intermediate mental zone, and outer marginal zone. The cells of ependymal zone are germinative in nature. Progenies of the most of these cells migrate outward into the mental zone and differentiate into the neuroblast and spongioblast. The neuroblast cells gives rise to the neurons, whereas spongioblast cells give rise to the astrocytes and oligodendrocytes. The remaining non-migratory cells of the ependymal zone remain in this zone and forms the ependymal line of the ventricles as well as central canal of the spinal cord. The microglia cells are derived in marginal and mental zone from the blood histiocytes when blood vascular system grows around the neural tube. Mental zone forms the grey matter of central nervous system. This theory we discussed is known as classical theory of the histogenesis. We will now discuss the recent theory. According to recent theory, the neuroepithelial cells initially are elongated and extend from external limiting membrane to the internal limiting membrane. The nuclei of these cells lies close to the external limiting membrane and zone of DNA synthesis lies just next to the external limiting membrane, whereas inner process of this cell is attached to the internal limiting membrane. Neuroepithelial cells gradually become short in length so that nuclei occupy the intermediate zone. Afterwards, neuroepithelial cell becomes rounded, nuclei come near the internal limiting membrane. At this stage, neuroepithelial cells undergo mitosis. Mitosis can take place in two ways, horizontally as well as vertically. If mitosis takes place horizontally, the newly formed cells elongate through the intermediate and synthetic zone, and ultimately after that this cell will again become short, become rounded and come to the mitotic zone, and the next cycle of the mitosis is repeated. If the mitosis takes place vertically, the cells of the outer zone migrate outwards and differentiate into the neuroblasts and spongioblasts. Neuroblasts will give rise to the neurons and spongioblasts will give rise to the neuroglial cells. Progenies of the inner cells remain attached to the internal limiting membrane and form the ependymal cells.